Welcome to Mootechie. Today we are going to learn about a very fundamental process in biology, DNA replication. In this session, we will understand this topic step by step. Now, let us look at the visual introduction to DNA replication. The core concept here is very simple. DNA makes copies of itself. This copying process, which we call DNA replication, is all about duplicating our genetic information. Why do we need to do this? It is an essential preparation step for cell division. You will see terms like unzipping and the two strands, which we will explore in detail. So why is DNA replication needed? The core idea is that replication ensures the continuity of life. When a cell divides, through processes like mitosis, it needs to pass on its genetic blueprint accurately to the daughter cells. This is what we mean by genetic continuity, preserving information. It's also crucial for growth, as building new tissues require new cells with complete DNA. You can imagine a parent cell before replication, and after replication, it ensures that daughter cell A and daughter cell B both receive an identical set of instructions. Now, let's get into the basic idea of how replication happens. The core concept is that each strand of the original DNA acts as a template. The existing strand guides the formation of a new, complementary strand. How does it guide? It follows the base pairing rule. Adenine pairs with thymine and cytosine pairs with guanine. So, by reading the template strand, the machinery knows exactly what new nucleotide to add, building a brand new complementary strand. An important characteristic of this process is that it is semi-conservative. What does that mean? It means that in each new DNA molecule, one strand is the old, conserved parental DNA and the other strand is newly synthesized. So, the original DNA is half conserved. You end up with two daughter molecules, each containing one old and one new strand. This complex process is carried out by a team of special enzymes. Three key enzymes work together. First, Helicase. Think of it as the unzipper. Its job is to unwind the double helix. Then, DNA Polymerase acts as the builder. It adds the new nucleotides to build the complementary strand. Finally, Ligase is the gluer or the sealant. It joins the DNA fragments together to form a continuous, complete strand. Let's look at the role of Helicase first. It acts like a molecular zipper. This enzyme is responsible for identifying the starting point, called the origin of replication, and initiating the process. It moves along the DNA, breaking the weak hydrogen bonds that hold the two strands together. This separation creates a Y-shaped structure that we call the replication fork. Next is the master builder, DNA polymerase. This is the primary enzyme that synthesizes the new DNA strand. It reads the template strand and adds the complementary nucleotides one by one, strictly following the base pairing rules. A very important point to remember is that DNA polymerase can only build in one direction, from the 5' end to the 3' end. This directionality has major consequences, as we will see now. This directionality leads us to the concepts of the leading and lagging strands. Because the two strands of DNA run in opposite directions, they are anti-parallel. The copying machinery has to work differently on each strand. One strand, called the leading strand, is synthesized continuously in the direction of the replication fork. The other strand, called the lagging strand, is synthesized discontinuously in pieces away from the fork. But remember, on both strands, synthesis always proceeds in that 5' to 3' direction. Those pieces made on the lagging strand are known as Okazaki fragments. Since DNA polymerase can only work in that one direction, it has to keep starting new segments as more of the template becomes available. These are short sequences, about 100 to 200 base pairs long in organisms like us. They are synthesized backwards relative to the fork movement, but still in that 5' to 3' direction. We are left with all these fragments on the lagging strand. This is where our third enzyme, DNA Ligase, comes into play. It acts like the molecular glue. DNA Ligase seals the gaps or nicks between the Okazaki fragments. It forms the final phosphodiester bond to create a continuous DNA backbone. So, from many fragments, we get a solid strand. What is the final result of all this coordinated activity? The outcome is two identical DNA molecules. We have achieved accurate, faithful reproduction of the genetic code. The cell now has two complete sets of genetic instructions and is ready to proceed with division, giving one copy to each daughter cell. Let's do a quick visual summary. Step 1 is unwinding, where Helicase unzips the double helix. Step 2 is copying, where DNA polymerase builds the new complementary strands. Step 3 is joining, where ligase seals the fragments on the lagging strand. The end result? Two identical DNA molecules, ready for cell division.